Greetings from Dr. SSO Tech YouTube channel. Today we have a look on HPLC, High Performance Liquid Chromatography, also known as High Pressure Liquid Chromatography. HPLC is an analytical technique used to separate, identify and quantify specific components in mixtures. The mixtures can originate from foods, chemicals, pharmaceuticals, biological, environmental and agriculture. This is an excellent 1260 infinity quaternary liquid chromatography. Principle involved in HPLC is the separation principle of HPLC is adsorption. It is based on the distribution of the analyte between the mobile phase and stationary phase. Depending on the chemical structure of the analyte, the molecules are retarded while passing the stationary phase. The specific intermolecular interactions between the molecules of a sample and the packing material define their time on column. Hence, different constituents of a sample are eluted at different times. Thereby, the separation of the sample ingredient is achieved. Let us see the components of HPLC. This is solvent cabinet. The solvent cabinet is a case to keep 4 bottles with solvent standing frame. An HPLC system begins with the solvent reservoir which contains the solvent. The solvent should be filtered with an inlet solvent filter to remove any particles that could potentially damage the system's sensitive components. Here, 4 solvent glass bottles or solvent reservoirs are present for isocratic or gradient analysis for rapid method development and also speed up preparation of mobile phases and flushing the HPLC system. They can be connected by solvent tubings to the LC system. In excellent 1260, degazing of solvents is done in a built-in vacuum degazer. This is pump. Here, quaternary pump is used. The pump generates a flow of eluent from the solvent reservoir into the system. High pressure generation is a standard requirement of pumps. Here, pressure range is up to 600 bar. In HPLC, 3 common types of pumps are used. They are constant pressure pumps, syringe type pumps and reciprocating piston pumps. Here, pump used is reciprocating pumps. This is solvent purging. It is to remove contaminants, air bubbles and ensure equilibration in the entire chromatographic system. This is solvent or mobile phase outlet. This is thermostated column compartment. It is temperature controlled column compartment is used for heating and cooling in order to meet the extreme requirements of retention time reproducibility. Separation of sample is performed inside the column. Stainless steel columns are most commonly used. The packing material generally used is silica or polymer gels. It is C18 column, probably the most popular type of column for HPLC. Why is the C18 column used in HPLC? Because of the extra carbons, C18 has a large surface area that the mobile phase has to travel across. This offers more interaction time between the bonded phase and the elutes. Thus, the sample elutes more slowly and has more separation. It is C8 column. It has octylsilane as its stationary phase and this compound in the stationary phase has 8 carbon atoms in its alkyl chain. Why is C8 columns used in HPLC? C8 HPLC columns are suitable for the analysis of a large number of non-polar substances and many ionizable and ionic compounds. It is GOT column. The GOT column is installed between the injector and the analytical column. It is designed to increase the lifetime of an analytical column by protecting the analytical column from unwanted materials such as highly retained or irreversibly retained compounds and particulate matter. It is detector. Separation of analytes is performed inside the column, whereas detector is used to observe the obtained separation. The composition of the eluent is consistent when no analyte is present, while the presence of analyte changes the composition of the eluent. Hence, detector measure these differences. This difference is monitored as a form of an electrical signal. A common HPLC detector is absorbance detector, such as ultraviolet absorption detectors and photodiode array detectors. Here, diode array or variable wavelength detector is used. It is manual injector. Manual injection consists of a rotary valve, also called rheodyne injector, with two main positions, load position and inject position. When the valve is in the load position, the sample can be introduced in the system with a syringe. Sample is introduced using microliter glass syringe. Entire HPLC system is connected to PC. In Agilent 1260 Infinity HPLC, software used is OpenLab software. This is the entire HPLC system. Applications in HPLC. HPLC in pharmaceuticals. The pharmaceutical industry is one of the biggest users of HPLC to check the purity and consistency of the products. They also use it to evaluate formulations, separate out compounds to be used in the production of new drugs, characterize molecules that can be used to manufacture new drugs. HPLC in clinical research. HPLC is a highly accurate method for analyzing the nutrient composition of blood, analyzing urine and blood for the presence of antibiotics, analyzing bilirubin and biliverdin analysis for hepatic disorders, analyzing endogenous neuropeptides in cerebrospinal fluid. HPLC in food safety and food quality. Analyzing samples to determine chemicals used in agriculture and food production, analyzing pesticides and food additives in food products, determining the quality of water and soft drinks, sugars in fruit juices, polycyclic compounds in vegetables. HPLC in environmental. When it comes to environment, HPLC is useful in the biomonitoring of pollutants and pollution levels and detection of phenol compounds in drinking water, and also environmental samples can include wastewater, drinking water, soil, sediments and many more diverse matrices. HPLC in forensic applications. Investigation of doping and athletics, identification of anabolic steroids in serum, urine, sweat and hair, forensic analysis of textile dyes, determination of narcotics in blood and urine. HPLC in nucleic acids research. In nucleic acids research, HPLC instrument has vital importance for some purposes like studying the regulatory effects of cyclic nucleotides, determination of the composition of hydrolysates of nucleic acids, metabolite profiling of normal and diseased subjects, separation and purification of nucleic acids.
Thank you. For more videos, like, share and subscribe the channel.